previous resources, the process also cost about $2.5 billion as of 2013. You can see in this chart that the cost has been increasing over time for both the pre-human as well as the clinical trial. So our big question is, how do we reduce the time the molecules tested and the cost? And we define that each set of 30,000 abstracts or 1 XML is 1 partition. And then we make it to where all of our functions work sequentially with pandas data frame as well as in parallel with DAS data frame. And now I will pass on to Jerry for our next step.
After that, we have to find 2 parameters, the K and L. The K and L are numbers of row clusters that stands for the drug and gene pairs, and L is the number of column clusters that stands for the dependency paths. We follow the heuristics created by the original paper.
Final presentation of the MLBI Team 2. I'm going to start by highlighting the team members. We are a team of 6 members. I'm the project lead, I'm Wafa, and Rachel is the project management lead, and we have 4 members.
It uses the OS and the glob Python libraries by looping through a data directory and creating key value pairs based on the files present. Then those pairs are added to the graph using the update function.
I'm in charge of the EBC, which is the ensemble by clustering for classification algorithm. And there's 2 steps to this. There's an unsupervised step and then a supervised step. So for the first unsupervised step, you use a certain clustering method called ITCC, and that bi-clustered that dependency matrix that we had earlier, the one that had rows of drug gene pairs and columns with dependency paths.
To get the co-occurrence statistics, module 4 is to get the sampling and the supervised step of the EBC algorithm, and module 5 is to actually get our results and the final dendrogram of the final clusters.
I mapped all the drug genes with their respective drug gene pair cluster and the dependency path with their respective dependency path cluster assignment, and created this data frame.
This decision is asking, is the test set co-clustering with the seed set? If yes, and if that's happening more frequently, we are assigning a high ranking. And if no, we are assigning a low ranking. So we're prioritizing methods that score the seed set knowledge high up.
On the right is the same sentence as in the last slide. And given the drug name and the gene name, we can find how the 2 words are connected to each other and their relationship with other words, like they're connected to decrease, and decrease is like the KM is the thing that is decreased, and then TH is connected to KM.
Now, to get a certain number of clusters, we need to cut the dendrogram at a particular height, and that's what we do here. In the end, we plot the dendrogram along with the cluster assignments, the existence of drug gene parents, pharmGKB, and drug banks, and bars accounting for frequency. So these are the final dendrogram plots. The one on the left is a standard dendrogram tree. We have cut the dendrogram at a height of about 0.74 to get 25 clusters assignment.